Welcome to Riffyn. In this video, we will illustrate Riffyn's use for a drug metabolism study in large animals. We will begin by logging into the Riffyn cloud via our web browser. This takes you to Riffyn's process library. A process can be any research activity, from cell development to screening, bioprocessing, analytics, purification, food processing, or basically anything else in your research program. The library includes both processes created by you and by other people who have shared those processes with you. You can have different roles: administrator, operator, designer, or viewer, and you can control who has access to each of your processes. For example, here, the cell culture process, we can open the share window and see who else has access. Here, 18 different people or groups or teams have been provided access at different levels of access control. The processes can be shared, made shareable, or public, and you can add anybody to a process. Here you see organizations, teams, and individuals that can be added to this process. Now let's open up the animal study. This takes you to the process designer. Here we enter design mode. In design mode, you see the processes that you can create, which are methodological templates for experiments you execute. Think of each process like a visual protocol or SOP. Here, for example, you see all the steps in the animal study, and you see groups of steps, such as the analysis group. Every process allows you to define all the material inputs and outputs of each step and track them across from step to step. For example, in this formulation step, we see the API compound and the vehicle and technician as inputs, and the output is an API solution, along with the properties that will ultimately become the data fields for your experimental measurements. You can also add new steps, and on each step you can add new inputs or outputs, which you can look up from our material, resource, and equipment library, which contains over 80,000 terms in a controlled vocabulary. For example, we could add this compound, and then we can add properties to that compound, which also come from a controlled vocabulary, including the units relevant to that property. You can also add formulas to each property, which calculate the value of that property once you add data to it from other properties. Everything you see in a process is also version controlled. If we look here on the right, we see the version history, and if we go back in time, we can see what the process looked like in these earlier versions. When we return to the current working version, this will be snapshotted when you start an experiment and forms the basis for each experiment you execute. But if you want to, you can go back in time and execute new experiments on old versions. You can also annotate your processes with pictures and video. As you see here, this particular step has a video of how to inject intravenously into an animal. So this part of the process provides instructions to operators during the execution phase. Now let's take a look at an experiment. Experiments are listed in the Experiments menu, which contains a list of all experiments that are ever executed on this process. That's important because it allows you to collect all your data and analyze data from your entire experimental history all in one place. So we'll take a look at this one experiment on this process. Here we enter measure mode, which is where you can execute experiments on a process and collect data. For example, on step 1, we can enter and view the details of the study, including the investigator and the compound used. On step 3, we can see the animals that are going to be treated in this study and their characteristics, such as diabetic or non-diabetic type. On step 2, we can see the formulations that are being prepared to treat the animals, in this case 2, an oral and intravenous formulation. And we can also see how these different formulations are going to be fed into the different of the 12 animals. For example, if we select this first formulation, it highlights the first 6 of the animals in the dosing and feeding step, because that's the formulation that's going to be used for those animals. Moving on to the analysis group, we'll now upload the compound concentration in the blood to the blood sampling step. We first select the step, we then go to the Record Data tab, and we click this file icon to collect data from a file. We select our file, and this takes us to the file parsing configuration utility. We select a previously configured file parser and open that up, which allows you to see the configuration for that file. Here we're parsing a block of data in a matrix format. These are the parameters for specifying the characteristics of that data block. These are the rules for assigning the data, and here are the rules for assigning the data to the properties. Here, for example, we're assigning the concentration field from the data file to the concentration property of the blood specimen. We can now upload and save that data, and if we view this now, we see that there is data on each of the animals treated in this study, and we click on the icon, we can see the actual data that's been uploaded. There's a time series over a 24-hour period for each of the animals in the study. At this point, we may wish to take a look at that data. We can plot it in Riffyn, selecting, for example, the sample time versus the API compound concentration on a linear or logarithmic scale, and we can color code it by aspects of the experiment, the mouse identity, for example. We can also share that plot if we wish, or even more powerfully, we can export all this data into a data file where all of the properties and data and settings from across the entire experiment are linked together into one file ready for statistical analysis. Example data. We type in a file name and export the data. That then processes and links all the data across the experiment. We can download that file and we can open that up in our favorite utility. In this case, we've opened up the data, the example data in JUMP, which is a statistical analysis program. All of the properties across our entire experiment are listed as column headings, and all of the rows represent the different samples and data points throughout that experiment. I've previously analyzed this particular data set with an example, which I show here. This is the exact same data but already previously configured for analysis. And we can take a look at the data as a time course. This is the same time courses that we saw in Riffyn, but now plotted in an external program. Or we can go further and we can actually ask questions of correlation across the properties of the animal and the treatment routes and other factors in the experimental study and how they affect the outcome of blood concentration in the animal. For example, we see here that the type of animal, diabetic or non-diabetic, significantly influences the concentration of this compound in the blood. But the gender does not matter, and the dosing route certainly does have an influence on the outcomes of blood concentration in this animal. This is all also controlled for the time point. This type of statistical analysis is very easy with Riffyn because all of the underlying data across that entire experiment is prepared and exported in a statistical data table that can be opened in any of your preferred analysis programs. That concludes our example, and I thank you for having a look at the power of Riffyn to drive your science.